Thank you for the invitation. Today I'm going to tell you about a project that's a collaboration between my lab, including Celia Blanco. Some of you have met her here. She's here at AbSciCon, as well as Robert Pascal's lab. And we have also essential input from Uli Muller and Jerry Joyce. So I'm going to tell you about this project, which was published a few months ago. Okay, so we are interested in the RNA world and in evolution of the RNA world. So for those of you who haven't thought about the concept of fitness landscapes before, let me just explain to you what we mean. So suppose you have a list of all the possible sequences, so 4 to the n, in our case n is 21 random nucleotides. You write them down in some fashion on a sequence coordinate. If you know their fitness, if they're ribozymes, maybe you know their catalytic activity, then you can write down this function in sequence space, and this function is known as the fitness landscape. On this landscape, the peaks correspond to families of active molecules. These troughs correspond to valleys of inactive molecules. So natural selection can be thought of rigorously as a random walk over this landscape. So a population will start out in some area of sequence space, exploring the local area through mutations. If a mutant arises that has higher fitness, then the population as a whole moves upwards, and so the population kind of climbs up the fitness landscape. So we think of it as a random walk with a bias toward climbing hills. So it looks nice on this cartoon, but we actually have very little idea of what real fitness landscapes look like. And we have some ideas from work exploring relatively local areas of fitness landscapes, looking around a known ribozyme, for example, and evolutionary pathways around a single known ribozyme. But what my lab is interested in doing is mapping this entire fitness landscape for the entire space. So of course we're limited by experimental sizes, so we can only look at relatively short sequences. Fortunately for RNA, even relatively short sequences, 21 mers, or at least sequences with a random region of 21 in length, can still be functional. So our experimental approach is based on in vitro evolution. So we take a pool of random sequences, many copies of each possible sequence, and then we subject this to a biochemical selection for the ribozyme activity we're interested in. The idea is that we'll reduce the frequency of sequences that lack this activity and enrich for sequences that have this activity. And in this way, we can pick out the active sequences. Okay, what are we interested in terms of activities? We're curious about the genetic code, like many of us here, and there's a lot of focus on the ribosome, and justly so. But the other part of the genetic code is the hooking up of the proper amino acids to the proper tRNAs. And sometimes that's called the second genetic code. So in modern biology, this is done by synthetases, the aminoacyl tRNA synthetases. Michael Yaris, many years ago, showed that it's possible to find ribozymes that can catalyze this reaction. So ribozymes that attach amino acids to RNA. However, before we jumped into the selection, we noticed that there are some problems which are experimentally difficult to deal with. This aminoacyl adenylate, the reactive substrate, is highly reactive in water. So it's not thought to be prebiotically plausible because of this, and it's also, from a practical standpoint, difficult to use in the lab. So we looked to our organic chemistry colleagues, Ziwei Liu, working in Robert Pascal's lab at the University of Montpellier, and they had come up with this form of activated amino acids, the 5,4-H oxazilones. These are related to what might be more familiar to you, the N-carboxy anhydrides. You can see, looking at the structure, the part that will become the amine and the carboxylate and the side chain of the amino acid. This pending group here is convenient because we Ziwei can put a biotin on there, and that way we have this capture handle for reactions. Ziwei had also noticed that these oxazilones, which are prebiotically plausible, they're formed from simulated volcanic mixtures. These oxazilones do react slowly with nucleotides. So in this case, I'm showing a modified tyrosine, which is very slowly reactive with nucleotides. So it's a perfect setup for a selection. We know the reaction is thermodynamically downhill. We want to find a ribozyme that catalyzes this reaction. So the selection scheme that was devised by Abe Pressman in my lab started with a DNA pool covering sequence space, transcribed into RNA. Most of these RNAs have no function that we're interested in, but a few of them will react with our substrate. If they react, they become biotinylated. By the way, this is biotin. So these biotinylated RNAs can then be captured by a streptavidin bead pull down. And then we can amplify these by RT-PCR, do high-throughput sequencing, follow the fate of these sequences over time. So that process gives you a list of active sequences. So think of this as a list of ribozyme sequences, which you've kind of culled from the space of all possible sequences. Then our next step, that what we really want to do is associate a catalytic activity, a rate constant, let's say, with each of those sequences. So to do this, we came up with a method with input from Uli and Jerry called kinetic sequencing. The idea is that we take a RNA pool which has medium diversity. It's not so diverse that we can't get much information about any particular sequence, but the pool has not converged so much also to the point where we only can look at a small number of sequences. So it's a medium diversity pool. We split it into several aliquots, react it with different concentrations of our substrate. You could also think of doing this with different time points, but substrate concentrations were easier for us for technical reasons. And then we react them and capture them, capture the reacted molecules, sequence those captured molecules, and then that gives us basically 4 points along this kinetic curve for different concentrations, and that allows us to calculate the rate constant for these sequences. And depending on how deeply you sequence, you could get information for thousands to perhaps hundreds of thousands of molecules. This kinetic sequencing scheme works pretty well at predicting the activity. Here's a correlation between the measurement by K-seq, kinetic sequencing, and a traditional gel shift assay. Okay, so now we have this list of ribozyme sequences and their associated rate constants. What we want to do is answer some interesting questions about this fitness landscape. One interesting question is, how smooth is this landscape? And the reason why this is interesting is if you had a very smooth landscape, as kind of an extreme example, a single peak, very smooth landscape, then you can imagine no matter where you start on this landscape, the population can feel the peak. So you can do a very smooth walk up to the top and find the global optimum over sequence space. On the other hand, if you have a highly rugged landscape like this, then kind of depending on where you start in the sequence space, you might make a local climb to what you think is the top of a peak, or what is the top of a peak, but you've missed the global optimum. So we were very interested in understanding these potential evolutionary pathways. Here's an example of the type of pathways that we find. So over here is one motif, motif 1 of the ribozymes that we found, ribozyme 1b marked in blue over here, motif 2 over here. You can see motif sequence 2.1 is the global optimum of the landscape. So what we can observe is that looking at the activity of these sequences, which we measured by K-seq, and the evolutionary distances, we can find pathways that connect relatively related sequences. You could call this motif 1b and 1a local optima, which are connected by reasonable evolutionary pathways. But going from this motif 1 area to motif 2, you have to go through a large valley of sequences that have basically no activity, or at least baseline activity. So this tells us that only nearby peaks are connected by viable pathways. If we zoom out to the entire landscape, so I'm not showing all of the different ribozymes that we found. There would be thousands of points if that were the case. But what I'm showing you is the top motifs that we found, which are shown by these big circles, and the best 5 pathways that we found between these major ribozyme centers. So what you can see is sequence 2.1, high activity, the highest activity over the landscape. This kind of low-lying plateau, I would say, of motif 1, where there might be some reasonable interconnections, but you have to look a little bit hard for them, but they exist. But motif 2 in particular is kind of cut off from the rest of this landscape by multiple mutations. These dotted lines indicate multiple mutations required. Multiple mutations required at essentially no activity to traverse this part of the landscape. So with that, I'd like to just close by acknowledging the people involved in this work. Abe, who led this project, and Celia contributed to a ruggedness analysis that I didn't have time to talk about, and Evan contributed to some experimental validation, which I also didn't have time to talk about. And I'd like to again thank our collaborators, Ziwei and Robert, and Uli and Jerry. Thanks very much.
I think we have time for one question.
That was a really interesting talk. Thank you. I just wondered, in terms of building up your evolutionary landscape, whether you're going to build in looking at the possible role of mobile genetic elements, which could potentially cause a discontinuity in evolution that could bridge some of those gaps.
Okay, yes. So we completely, I really oversimplified this this picture, saying that we're just looking at these step-by-step mutations. We have in our pathfinding algorithm, you can allow larger jumps in sequence space, and you get pretty much the same picture if you allow up to 4 mutations. But if you allow quite large gaps, then you can start to see kind of a completely interconnected network.
Okay, let's thank Irene one more time.